Good morning, students. Today we will see hydrochloric acid secretion, HCl secretion or gastric secretion. The gastric secretion is occurring in phasic manner. There are three phases by which the secretion occurs. The first is the cephalic phase, which constitutes about 30% of the gastric secretion. Second is the gastric phase, which constitutes about 60% of the gastric secretion. And third is the intestinal phase, which constitutes about 10% of the gastric secretion. Among these three phases, the cephalic and gastric are excitatory phases, and intestinal phase is a minimal excitation, and it is predominantly an inhibitory phase. So this is the percentage division of the gastric secretion. So now we will see the each phases individually. The first is the cephalic phase. Cephalic phase, I told you 30% of secretion, and it is more of neural. So predominantly neural. It is regulated by the vagal efferents. So what happens when the food is exposed? When you see the food, the sight, smell, thought, and the taste of the food, it will activate the brain higher centers like the cerebral cortex and hypothalamus, and they send the stimulus to the vagal nuclei in the medulla, and through the vagal efferents, acetylcholine is released in the stomach. This acetylcholine acts on the parietal cell on its receptor, and it stimulates the HCl secretion. Also, the vagal will release a peptide, gastrin releasing peptide, that stimulate the gastrin cell, that is G cells, to produce gastrin. So this gastrin in turn acts on the parietal cell and stimulate the HCl secretion. So the cephalic phase is predominantly it is neural and partly the hormonal because of the gastrin release. The next phase is the gastric phase. Gastric phase constitutes about 60% of the gastric secretion and is a major phase. It occurs when the food enters into the stomach. So entry of the food causes distension of the stomach, and this activates the reflexes. There are two reflexes activated. One is vagovagal reflex, other one is the local myenteric reflex. So this vagovagal reflex, here the afferent is vagus nerve, which carries a sensation to the vagal center in the medulla, and through the efferent it activates or it releases acetylcholine, and it stimulates the parietal cell to secrete HCl. So afferent is also vagus, efferent is also vagus. The second reflex is the local reflex. It is a myenteric reflex. The distension causes activation of the vagal or the myenteric neurons, and that releases acetylcholine, and that causes the parietal cell to secrete HCl. So this is a long reflex, and this is a local reflex or a short reflex. So the gastric phase, the neural part is contributing for the gastric phase. The next part is the hormonal. So here the hormone gastrin is released on the distension. Not only distension stimulate the gastrin from the G cells, also it is stimulated by peptides, amino acids, and digested products of proteins. And this gastrin in turn stimulates the ECL, enterochromaffin-like cells, and that releases histamine, and both histamine and gastrin acts on its receptor in the parietal cell, and histamine acts on its receptor on the parietal cell, and they stimulate the HCl secretion. So here the neural component is there because of the two reflexes, and you have the hormonal component So which is predominant in the gastric phase? It is the hormonal component, that is the hormone gastrin and gastrin induced histamine release. The last phase is the intestinal phase. It constitutes 10% of the gastric secretion. So that is the excitatory part, where when the protein digested products or the acid chyme, when it enters the intestine, it stimulates the duodenal G cells to secrete gastrin. And as I told you, gastrin acts on the parietal cell on its receptors and stimulate the HCl secretion. So this is hormonal. Excitatory is purely hormonal. Then inhibitory part. The inhibitory part is a major part. It has two components. One is the enterogastric reflex, other one is the enterogastrones. The enterogastric reflex is when the acid chyme or the products of protein digestion enters the intestine. So this intestine, through the enteric neurons, it will stimulate the sympathetic neurons, and these sympathetic neurons will inhibit the gastric motility and gastric emptying, and thereby the gastric emptying is delayed. So enterogastric reflex is a preventive reflex. It is like it delays the gastric emptying so that the digestion is proper, and it gives some time for the pancreas to secrete bicarbonate so that it will neutralize the acid chyme. The next part is the enterogastrones. Enterogastrones is purely hormonal. Here the stimulus is the same products of protein digestion and acid chyme that will stimulate the cells in the intestine, S cells to secrete secretin, K cells to secrete gastric inhibitory peptide, and I cells to secrete cholecystokinin. And these hormones have a negative influence. It will inhibit the gastrin release and thereby it reduces the HCl secretion. So this is by the release of enterogastrones, which is hormonal. So to sum up the intestinal phase, it is minimal excitatory is that is hormonal, and the major inhibitory part that is contributed by both hormonal and neural. So we have three phases of HCl secretion. One is the cephalic phase, gastric phase, and intestinal phase. Cephalic phase is purely neural, influenced from the higher centers through the vagal efferents. Gastric phase is neural and hormonal, and predominantly it is hormonal. And intestinal phase is a mixed phase. We have excitatory and inhibitory events, and excitation is by the hormones and inhibition by neural and hormonal components. Thank you.